At this juncture, we now proceed with radiation carcinogenesis. Ionizing radiation causes chromosome breakage, translocations, and less frequently, point mutations leading to genetic damage and carcinogenesis, as reflected on the illustration below. The diagrams elicited UV rays that induce the formation of pyrimidine dimers within DNA, leading to mutations. Therefore, UV rays can give rise to squamous cell carcinomas and melanomas of the skin. Individuals with defects in the repair of pyrimidine dimers suffer from xeroderma pigmentosa and are at particularly high risk. Imaging procedures such as CT scans is linked to a very small but measurable increase in cancer risks in children. Right at this moment, we talk about microbial carcinogenesis, which are usually brought about by oncogenic DNA virus and oncogenic RNA virus. There are 5 DNA viruses that have been implicated in the causation of human cancers. One of them is human papillomavirus or HPV. It is further subdivided into two. Low-risk HPVs cause benign squamous papillomas or warts by HPV 6 and HPV 11. Meanwhile, high-risk HPVs implicated in the genesis of squamous cell carcinomas of the cervix, anogenital region, and head and neck, particularly tumors arising in the tonsillar mucosa brought by HPV 16 and HPV 11. High-risk HPV types express oncogenic proteins that inactivate tumor suppressors, activate cyclins, inhibit apoptosis, and combat cellular senescence. Take note that oncogenic types of HPV encode two viral oncoproteins, E6 and E7, that bind to RB and P53 respectively with high affinity and neutralize their function. But don't forget about Epstein-Barr virus or EBV, which is the first virus linked to human tumor Burkitt lymphoma, a neoplasm of B lymphocytes that is endemic in Central Africa and New Guinea, areas where it is the most common tumor of childhood. It seems that EBV is not directly oncogenic, but by acting as a polyclonal B-cell mitogen sets the stage for the acquisition of the 814 translocation and other mutations that ultimately produce a full-blown cancer. Most EBV-associated tumors are commonly lymphomas derived from B cells and nasopharyngeal carcinoma. Nasopharyngeal carcinoma are endemic in southern China, parts of Africa, and the Inuit population of the Arctic. In the absence of the T cell immunity, EBV-infected B cells can rapidly grow out as aggressive B cell tumors. In the presence of normal T cell immunity, a small fraction of infected patients develop EBV-positive B cell tumors or carcinomas. Third oncogenic DNA virus is the hepatitis B virus or HBV and hepatitis C virus or HCV, which account to 70 to 85% of hepatocellular carcinomas. HBV is endemic in countries of the Far East and Africa. Oncogenic effects are multifactorial. Dominant effect seems to be immunologically mediated chronic inflammation, hepatocellular injury, and reparative hepatocyte proliferation. HBX protein of HBV and HCV core protein can activate signal transduction pathways that may also contribute to carcinogenesis. Merkel cell polyomavirus and human herpes virus 8 or HHV8, also called Kaposi sarcoma herpes virus, are oncogenic DNA virus. The oncogenic RNA virus include human T-cell leukemia virus type 1, only human retrovirus, which is firmly implicated in the pathogenesis of cancer in humans. It causes adult T-cell leukemia or lymphoma or ATLL. ATLL is a tumor that is endemic in certain parts of Japan, the Caribbean Basin, South America, and Africa, and found sporadically elsewhere, including the United States. HTLV1 has tropism for CD4 T cells, and hence this subset of T cells is the major target for neoplastic transformation. The genome contains regions typical of all retrovirus, but in contrast to other leukemia virus, it contains another gene referred to as TAX. TAX is the protein product of this gene. TAX contributes to the acquisition of several cancer hallmarks to include increased pro-growth signaling and cell survival and increased genomic instability. Helicobacter pylori is also implicated in gastric adenocarcinoma and malt lymphoma. The pathogenesis of H. pylori induced gastric cancers is multifactorial, including chronic inflammation and reparative gastric cell proliferation. H. pylori pathogenicity genes, such as CAG-A, may also contribute by stimulating growth factor pathways. Chronic H. pylori infection leads to polyclonal B cell proliferation that may arise to a monoclonal B cell tumor or malt lymphoma of the stomach as a result of accumulation of mutations.